Hello, everyone. Thank you for attending the Bionano 2026 Symposium. It is my pleasure to speak today about high throughput structural genome analysis and scaling for the future. My name is Dr. Adam Smith, and I'm a clinical laboratory director for Labcorp out of the Center of Molecular Biology and Pathology in North Carolina. Okay, I think during this week, we've all heard a lot of really great things about OGM and all the different things it can do. And I wanted to take a moment and just talk about a couple of really significant milestones and the dramatic changes that have happened over the last couple of years. I started using OGM in our development of it in 2021 at the University of Toronto and the University Health Network. Over that time, there has been a tremendous amount of publications showing concordance of OGM to conventional cytogenetic techniques. We've seen CPT codes launched for hematologic and constitutional applications. Also, we've seen new instruments come onto the field. So the Stratus has come out giving increased throughput with more flexibility to run samples individually or more samples at a time, and using different run times. And also emerging now, we've seen recently automation options to help with the extraction of ultra-high molecular weight DNA and labeling from several companies. So when we start to put that picture together, what we really think, we really start to think differently than we thought from a few years ago. And I think the question is that we're asking ourselves now, instead of should we do OGM, it's how many can we actually do? Before we go into talking more about scaling, let's take a step back for a second to 2022 when we published this review paper about guiding the global evolution of cytogenetic testing in hematologic malignancies, and of course, more generally across cytogenetic testing at large. The impetus for this paper was really a technical report about the possibility of using short-read whole genome sequencing to replace cytogenetics. And what we said is that while the adoption of any new technology is not necessarily a question of whether or not that technology can detect the things that it's looking for or not, but it also needs to be in the laboratory context practical from a technical, logistical, and financial consideration. So there's all those considerations that are very significant that need to be thought about before any new technology is put into use in a laboratory. And so, we ask ourselves lots of questions from the feasibility perspective. And a very short list, of course, would be things like when we look at assay performance, we want to look at concordance with conventional testing. We want to see if there are community recommendations. We will look for things like proven utility. And so when we look back, over the last few years in the literature at optical genome mapping, we can certainly see that there have been lots of publications dealing with concordance, recommendations, and utility for OGM. But when we bring it into the laboratory and we want to incorporate it in any workflow, we also need to think about those technical and logistical things. So we need to think about can we automate sample processing, or is the workflow for sample processing reasonable? What changes to lab workflow is it going to mean? Are we going to displace some current testing with testing that we bring in? And then the other questions are things like when we get to the logistical side, how much custom work do we need in terms of bioinformatics? In the case of OGM, we have an end-to-end solution, so that's not really an issue. But we still need to figure out how we're going to put it in the lab and implement it and back up the data and do all the other things that we need to do. And then, of course, we have to think about interpretation and reporting, because at the end of the day, a scientist has to look at the data and try to figure out what's going on and write some kind of report. And of course, we need to do this in a method that is conscious of everyone's time, because this interpretation step is, of course, can take up a lot of time potentially. And this is very, can be very challenging in terms of, we only have so many hours in the day. And then ultimately, in terms of the financial considerations, it has to be a reasonable cost per sample. And then what are your ancillary costs in terms of, how many, servers do you need? How much data storage do you need to keep all of the data, raw data from all the assays that you're running? So again, it's not a simple equation in terms of how, just whether or not the instrument can do this one thing, like detect the type of copy number variation or structural variation. And so talking about the feasibility considerations, when we look for community recommendations, it's nice that we have started to see groups publishing on specific recommendations for testing that can be used. And so this was a publication from the International Consortium, which I'm a part, which you can go and look up if you want more information about all the different recommendations and where OGM can be used. I'd also like to highlight a paper that just recently came out at the beginning of the month in prepress format in the Journal of Molecular Diagnostics. And I'm very proud of the group in Vancouver in Canada, especially including my very close colleagues Tara Spence and Eric McGinnis, who did this really wonderful study looking at the clinical utility of optical genome mapping in these 200 cases. So they did this parallel testing on 200 cases, and they looked at the reassignment of the karyotype results versus OGM and whether or not there were additional findings. And so, what's obvious to pretty much all of us is, of course, that there were several additional very interesting findings that changed not only classification, but also risk. And when we actually enumerate those, what we can see is that in this cohort of 200 patients, we have 18% of samples that have either a change in their risk status or in their classification, sort of the type of leukemia classification based on the genomic abnormalities that were detected. And this is actually a really high number for a single technology in the genomics space. So let's say you've decided you want to bring in optical genome mapping testing, and we want to sort of look at the bottlenecks and where they are. So we have a bunch of different steps that we go through when we do OGM, and if you're familiar with OGM, you know this very well already. But so we have our wet lab processing, and you're going to have limitations imposed on what you can process by limitations of how much technical staff you can assign to do this, whether they work Monday to Friday, whether they work one shift a day or multiple shifts, or if you have 24/7 operation. You're going to have instrumentation bottlenecks potentially depending on the number of instruments you have, how many samples per week you want to run. And so here for the two current instruments that Bionano offers, you have the Sapphire and the Stratus. You can see there based on a 5, 6, or 7 day run schedule per week, you can see how many samples approximately per week per instrument you can run. Then after you've got all that great raw data, then you have to push it through your compute server and you have to generate assemblies. And so there's a limit, obviously, to what you can do with a single compute server. So then you have to think about all of that. Now you can add additional servers to process more data. That's no problem. And then, of course, at the end of the day, you've got to take all that data and you've got to interpret it and you've got to look at it. And so how we're doing that, there was, of course, there's a great paper in medRxiv published by Gordana Raka and colleagues, also a member of the ICOMG, and looking at using VIA for interpretation and how quickly using the decision trees and other tools in VIA for OGM analysis, you can get from first analysis out to a report. And this is sort of from the paper, of course, the first pass analysis is about 30 minutes from the Raka et al. Paper, but just putting in a few additional items on my own, we're ballparking sort of around an hour and 10 minutes a case, potentially. So again, these are all important factors to be able to put into a package when you're thinking about how much time and people am I actually going to have to use in order to get this going in the laboratory. And depending on the size of laboratory, you might think about what it is, in fact, that what you really need. And there's a bunch of different ways that you can do this depending on your annual volume. So this is just this table basically just gives you a breakdown about, if you're thinking about testing 1,000 or 2,000 or 4,000 or 10,000 samples a year, Basically, you can take these you can take a combination of instruments, or you can take a number of Stratus instruments, and you can process a lot of samples a year. 10,000 optical genome map samples a year would be a pretty high number, I think, for anywhere. But this is actually not really something that, from an infrastructure point of view, is that difficult to do. So, at 10,000 samples, if you have 4 Stratus or Strati potentially, depending on how many days a week your lab staff is working, you can batch those up. So for example, for 10,000 samples a year, we'd need 16 Stratus runs a week, and we need about 9 to 10 computes, depending on, I'm told there's going to be some improvements, again, time improvements in the compute pathway. So this might be less, even 6 months from now. It's hard to know exactly what it's going to be. But as per sort of our current experience with running guided assemblies, this is the kind of compute requirements you need to run 10,000 samples a year. So again, depending on the size of your laboratory and what you want to run, you can see here there's a way to combine instruments to do that. And the other thing I just wanted to put on your radar as well is that a lot of times when we have this set up in the lab, we think about redundancy. So it's, you might be able to get away with a Stratus for a particular volume, but sometimes 2 Sapphires, because you have 2 machines, there's redundancy in case you don't want to go right to 2 Stratus, or a Stratus and a Sapphire, for example. There's a bunch of different ways that you can combine the instrumentation to give you the redundancy that you need in order to keep running every day. On the sample processing side, of course, many of you are probably very familiar with the sample processing for OGM, and I don't need to belabor the process. There are some exciting developments here, of course, in terms of going from extracted DNA and then to labeling. So, of course, the manual processing isn't particularly laborious from a manual standpoint. However, when you start thinking about processing 2,000, 4,000, 10,000 samples a year, then you start to think about, well, maybe I don't want to put my entire wet lab team on doing this, and I want to try to automate it. And this, you can really reduce the amount of hands-on time that you're actually committing to the process if you're able to do the automation. And some of the new automation technologies, especially the ionic DNA extractor that Bionano is selling, also has a huge advantage in the DNA homogenization area because of the technology. And I'll just talk about that really briefly. So isotachophoresis doesn't actually take these DNA molecules out of solution like conventional DNA extraction technologies do. And it uses this combination of different buffers as it pulls the DNA out to its own specific band, which can then be taken out and pretty much moved into a labeling reaction with almost no delay, about an hour or so. So this also takes a huge amount of time out of the entire wet lab process. And when we start to talk about analysis, what we've also seen, of course, is that when we started doing this, we did this all in Access, and we had a nice little process that we set up where we were collecting PDF summaries of the data just after applying basic filters, so that we got all of the structural variants, so we could go back and always look at that. Then another PDF with our curated variants, and we did some additional filtering on specific gene regions that we were interested in, say for hematologic malignancies, and then we could very easily from that curated data generate a report. However, very time consuming, very manual, lots of cutting and pasting, and not very automated. And of course, recently, as I talked about the paper published by Raka et al., by using VIA now, there is a process whereby this analysis can be streamlined with decision trees and other things that will really speed up the process of collecting all of the important variants that we're looking at and summarizing them, outputting them without having to do a lot of manual data entry right into a lab director's queue for review, and then they can be reported easily. The other nice thing about VIA is that it can be deployed, in larger installations or larger institutions with a cloud-based VIA server. So this is one of the things that we're looking at, where we have multiple lab sites. They're using multiple technologies that can actually all upload into a cloud-based VIA. And then wherever our analysis team is, they're distributed all over the place. They can log in and access VIA, do the analysis, and keep the whole process of interpretation moving. And the other nice thing about this for us is that we take all of our different pathways, we've harmonized them into one analysis pipeline, so that all of our interpretive scientists and technologists and directors are all using the same software and tools, no matter what the technology for detecting copy number or structural variants, which is also very important for keeping consistency. And so just to end, I just wanted to point out that, we've seen great promises, as I talked about, from genome sequencing as an alternative to the analysis of myeloid cancers. Of course, this was published in 2021, I believe. And so it's been a few years. And then the same group actually published a follow-up to this using long-read sequencing, where now they say long-read's much better. But just before we really dive into that, the things that we talked about at the beginning in terms of things like, well, where are the community recommendations? Where are the concordance studies outside of a few individual papers? And we really haven't seen them. And I think if we take a minute to really think about the whole picture of when we try to implement these kind of technologies, this is sort of where we get to. So let's take, for example, a lab that wants to run 1,000 samples a year, and they're going to do PacBio Revio. And I put a lot of details here that I've just basically got out of general publications from the PacBio site or off of the web. So we could argue about the exact numbers. That's not really the point. The point is, if you try to do 1,000 of these a year on a PacBio Revio, you're going to need 4 to 5 of these at least. Okay, that's if you don't want any redundancy. So you're talking about a huge capital investment of several $1 million before you even invest a cent in bioinformatics. On the other hand, a single Stratus instrument will do 1,000 genomes without breaking a sweat. So these are the kinds of considerations we always need to keep in mind whenever we look at implementing new technologies in the laboratory. There's nowadays lots of options for doing structural variant detection. They come with their own advantages and disadvantages, as you can see here from the philosophical approach to analysis. The karyotype and OGM take a more, I'm actually looking at the data type of approach. With the karyotype, you look at chromosomes. With the OGM data, you can actually look at the molecules, you can see it. It's not exactly the same. I'm not suggesting they're exactly the same, but it is different than the sequencing approach, where you're really inferring the presence of structural variants, and this can lead to other challenges in terms of data interpretation. To finish off, I'd really just like to make a plug for the International Consortium for Optical Genome Mapping. This is a group of lab directors and scientists that work from more than 18 countries across the world. We have 15 working groups in progress right now. It's open to anybody basically that's using OGM. If you want to join, go to the website, click on membership. It's free to join. And these are all of the different places that are collaborating into the ICOGM. It has been a fantastic group in terms of developing and sharing information about OGM. And really, I would encourage everybody, if you're using OGM, to sign up for the group and join us. And with that, I would like to thank you all for your attention, and I hope you've enjoyed the symposium, and thank you all for your presence.